Hello, my name is Akiko Iwasaki. I'm a professor of immunobiology at the Yale University School of Medicine. I've had my lab here for 22 years, and my laboratory studies immune responses to a variety of insults, mostly focused on mucosal infection with a variety of viruses, as well as more recently against cancer. I study the immune system because it is a fascinating and beautiful system where the immune system utilizes multiple different cell types that can travel from one location to another to provide both immediate defense against infection, as well as to provide long-lasting immunity so that you don't have to get infected with the same pathogen twice. The immune system remembers not only the type of pathogen, but also the location of where the pathogen was encountered. And by studying the mucosal immune system, we are able to elicit basic mechanisms by which cells migrate and remain in different tissues. And from that insight, we created a different vaccine approaches, including the prime and pull, which enables effector T cells to seed a particular tissue of interest, the mucosal tissue, where the virus normally encounters the host. And this prime and pull strategy can be utilized to block infection with herpes simplex virus, which causes genital herpes, as well as it has been shown to work really nicely as a therapeutic vaccine against recurrent genital herpes using a guinea pig model. And during the COVID-19 pandemic, we used the similar insights to apply our knowledge to the respiratory tract. And we developed a new vaccine strategy called prime and spike, which can be used to leverage circulating immune system, particularly the CD4 T cells, to stimulate a new immune response in the nasal cavity and in the lung by delivering the spike protein of SARS-CoV-2 virus intranasally, unadjuvanted. And this enables establishment of long-term immune responses in the nose and throat and the lung by generating memory cells that can be re-stimulated quickly upon re-encounter of the virus, as well as establishing IgA, secretory IgA that can bind and block the virus as they enter through the mucus layer, so we can potentially provide sterilizing immunity against SARS-CoV-2. And because it's a nasal spray format, it's also much easier to administer than an intramuscular vaccine. So the reason we study the immune system is not only to gain basic insights, but also use the basic insights to help others and hopefully save millions of lives. That would be our dream come true. Thank you for listening.